Assalamu alaikum, guys. So, what we were studying last time was the process of DNA replication. And if you remember, we have discussed in detail about the DNA replication process in prokaryotes. So, we're going to continue this today with our next discussion of the process of DNA replication in eukaryotes. So still you guys don't follow the time restrictions and then you guys complain that you are not being allowed to enter the class. I don't know what is actually preventing you guys not to follow the schedule time. So today I'm going to admit every one of you, but make sure that from the next class I'm not going to add you guys after the time that I have given. It has to be pretty much clear to all of you now. If you miss, you miss. You can listen to the recording because we don't want the interruption in the class. Okay, so what I was talking about is that we discuss about the process of DNA replication. We know that this is the property of each and every cell to replicate. And as a result of this, we need to multiply the genetic material. So this is a property that is exhibited by every living cell. So what we discussed in the last class was about the in detail process of DNA replication in prokaryotes. Now today we're going to start with the DNA replication in eukaryotes, and I'll try to finish this lecture today. Let's see how it goes and whether we would be able to complete this lecture today, inshallah. So just keeping in mind that the DNA replication process will definitely be more complicated in case of the eukaryotes, because when you compare the genetic material of a prokaryotic cell and a eukaryotic cell, it's quite clear that in case of the prokaryotes, you have studied that they have simpler genetic molecule. Their DNA is circular, if you remember, and they have few thousand genes on them. While in case of the in the eukaryotes, their genetic material is much more complex, much more, I would say, bigger, and it contains much more number of genes on it. And obviously, another difference was that the eukaryotic DNA is a linear molecule. So keeping in mind that point, that it's more complicated, more diverse, and more bigger in size, so definitely there will be more complication when it comes to the DNA replication in eukaryotes. So let's start with few of the basic information about the eukaryotic DNA replication. If you remember, in the prokaryotic DNA replication, we studied about ORI, which is called origin. And I mentioned to you guys that origin is a specific point on the DNA molecule which is being identified by particular enzymes called helicases. And the function of those helicases was to identify the origin they bound at that site, and as a result of that, a replication fork is generated. This replication fork is generated because of the fact that the hydrogen bond between the opposite nucleotide base pairs is broken down as a result of the binding of the helicases. And that's how there is the process of unwinding of the two strands takes place. Now, a very similar process in eukaryotes, because the origin is having the same concept as that was in the prokaryotes. The difference here is that in the DNA of the eukaryotes, you have multiple origins within the same molecule of the DNA. And the answer to that is the same as I said earlier. In contrast to the prokaryotic DNA, the eukaryotic DNA is much bigger. It contains thousands and thousands of genes on that. And that's why it has multiple origin sites simultaneously on one DNA molecule. On an average, the human genome is about having 30,000 origins. So you can see how complicated the molecule is, and look at the number of origins which are present on the human genome. So that's what the concept of multiple origin in case of the eukaryotes. Now, each origin will have the same purpose as that it was in case of the prokaryotes. What was the purpose of the origin? Origin gives a site from where the replication fork is generated. And this replication fork is, as I said, generated because of the breaking of the hydrogen bonds, you can see here. And this is being definitely done by a mechanism which you studied in prokaryotes, that helicases will bind to the origin and results in the breakdown of these hydrogen bonds. Thus, unwinding takes place, and as a result of this, the replication fork is produced. So the concept is same, but we have multiple replication forks in case of the eukaryotes because we have multiple origin sites. Similar to the prokaryotes, if you remember, they have a circular DNA, and I told you that the replication fork run bidirectionally, both clockwise and anti-clockwise. In a very similar way, the molecule here is linear, but you can see that the replication fork, they are the same. Like in prokaryotes, they are moving in opposite direction. If one is going to the left side, the other is moving towards the right side, and that's how the double helical strand is going to unwind. And you can see that as a result of the process going on, this is the intact eukaryotic DNA molecule. This picture is of the multiple origins you can see, and the multiple replication forks. And this picture shows that the replication forks are moving simultaneously in the opposite direction. And this final picture gives you two template DNA strands with the blue color, and two red nucleotide chains, which are the newly synthesized strand. And that's again, the concept that has been studied earlier on, which we call the semi-conservative replication mode. So are you guys clear the concept of origin between prokaryotes and eukaryotes, and the concept of multiple replication forks within the same DNA molecule? Now, here is a table which gives you an idea about the number of enzymes involved in the process of DNA replication in eukaryotes. And you can see that it's much, much higher in contrast to what you have studied in bacterial replication mode. If you remember, I told you about 5 DNA polymerases, and out of those 5 DNA polymerases in prokaryotes, the most significant in terms of like the process of replication, or I would say rather the enzyme which is involved in the elongation of the chain, was DNA polymerase 3. But here you can see in the human, there's a long list of different enzymes, DNA polymerases, which have their own functions. So I'm not going in, details of each and every of them. You can see that we have highlighted few of them, and the one that we have highlighted are the one that we're gonna discuss in detail, or rather, I would say that the highlighted one are the one for which we are pretty much sure about their role and their function. So it's polymerase 1, polymerase 3, and polymerase 2, which is being also called as alpha, delta, and epsilon. So, but beside that, you can see there are a long list of other polymerases, and their proposed functions are also given there, and in some cases the function is not yet well understood as well. So this is another indication of the fact that the DNA replication in eukaryotes is more complicated than the prokaryotes. Now, in case of the DNA replication in eukaryotes, now you have to be very careful that there are many basic sort of similarities. When it comes to the process of replication, there are uh, similarities between the replication in prokaryotes and the eukaryotes. But as a matter of fact, because the eukaryotes are more advanced, you would have more complication. You would have more extra processes involved when it comes to replication in eukaryotes. But the basics would remain the same, which we have already studied in the last lecture. So here are the enzymes which I mentioned, that in eukaryotes we have almost 15 DNA polymerases that have been identified so far. So what we are interested in, as I mentioned in the previous slide, polymerase alpha. This polymerase alpha, which is also being called as polymerase 1, will be acting as a primase enzyme. Remember, in when we studied about the DNA replication in prokaryote, we studied that the DNA polymerase enzyme is not capable of synthesizing the chain elongation of two nucleotides. It requires a small set of nucleotide to start reaction, and that set of nucleotide was called primer. And the enzyme that synthesized primer in case of the prokaryote was primase. Now it's the same enzyme, but with a different name. Here it's polymerase alpha, which will be working like a primase, and that is going to synthesize the primer. And we all know that the DNA polymerase main enzyme is going to synthesize, or it's going to elongate that primer by adding more nucleotide to the 3' end of the primer, and thereby the polynucleotide chain is going to be synthesized. Then we have polymerase beta, which is not highlighted because, as I said, that the one we are more interested in, the one highlighted with the yellow, polymerase alpha, delta, and epsilon. But quickly going through a few of the other polymerases, and I wanted to share what their specific role is. Polymerase beta is a specific DNA polymerase which is involved in DNA repair mechanism. And what sort of repair they do? Two specific type of repair: excision repair and gap filling. Let me give you a brief idea what actually DNA repair is. When we have the process of DNA replication going on, there are chances that there might be some cuts or breaks in the DNA molecule. So as a result of that, the DNA requires repair mechanism. And why that repair is important? Because we all know that the genetic information of our body is stored in DNA. If the DNA undergoes any change, it results in a permanent mutation. And as a result of that permanent mutation, when the next time DNA replication takes place, it's going to replicate the false DNA. So we want to keep the DNA intact. We want to keep the DNA in its original form. So as a result of any damage or any sort of change during the process of replication or transcription, we must repair that DNA. This is a very, very key process. And polymerase beta is the one that's involved in the repair of the DNA. Now, it performs the repair by two different mechanisms. One is called excision repair, and one is called the gap filling. I think gap filling is very, very easily to understood. Like when there is a gap by the removal of a nucleotide base, and that results in a DNA damage, then this polymerase beta is going to fill up the gap and rejoin the DNA molecule. Similarly, excision repair is like if something has been added wrongly in the DNA, that has to be removed from the DNA. So two types of repair, either excision repair where there is a removal, and the gap filling where there is some missing part. Now, the next one is the polymerase gamma. That polymerase gamma is involved in the process of replication and repair both. But this gamma is specifically the polymerase enzyme which is present in mitochondria. Remember, I told you in the lecture earlier when we were studying about introduction to molecular biology, that beside the nuclear DNA, mitochondria has got its own DNA. And there was a reason for that, if you remember, that mitochondria has to synthesize its own enzymes and proteins. So there is a separate DNA in mitochondria. So this polymerase gamma you can say it's specifically the polymerase enzyme of mitochondrial DNA, and it has the same property, like it replicates, it repairs, and it also does the proofreading mechanism. So this one polymerase gamma has got all the functions of replication, repair, and proofreading, because that exclusively is for the mitochondrial process of DNA replication. Then polymerase delta. So this one is the main second enzyme, the most important enzyme, I would say. This is the main DNA polymerase enzyme of eukaryotes. It has high processivity rate and also a high proofreading rate. Remember, I told you in the last lecture that chain elongation will always be in the direction of 5 prime to 3 prime, while the proofreading is always, most of the cases, is 3 prime to 5 prime. That means it's going in the back direction as well, to see whether the correct nucleotides have been incorporated or not. So this polymerase is having a high processivity and also has got a proofreading mechanism that runs in 3 prime to 5 prime. As a result of the proofreading, if this enzyme diagnoses a wrong incorporation of a nucleotide, which is done by mistake, then this has got exonuclease activity as well. So that means it not only identifies the wrong incorporation of the nucleotide, but it also removes that wrong nucleotide. And this polymerase is primarily responsible for the replication of the lagging strand. Remember, there is one leading strand on which there is a continuous synthesis of the chain, and there was one lagging strand where you've got fragment synthesized, what we call Okazaki fragments. Polymerase epsilon. This is again a highly processive enzyme and has also got the proofreading mechanism of 3 prime to 5 prime. It also has got the exonuclease activity, and this is the enzyme responsible for replication of the leading strand. So now you can see that in eukaryotes, it's a bit of a more complicated. Like in prokaryotes, there was one polymerase 3, which was, primarily involved for both strands. But here you have a separate polymerase for lagging strand. You have a separate polymerase for the leading strand. The polymerase for the lagging strand is doing proofreading, exonuclease, and chain elongation. So as the polymerase of the leading strand is also doing proofreading, exonuclease, and chain elongation. And both of them are highly processive. Highly processive means they have a good processivity speed. Now, beside that, there are a few other small polymerases, which I said that they might have different roles, but some of them have not yet been fully characterized, and their roles are not being fully defined. So I'm not going in the detail of those enzymes. So the DNA replication in eukaryotes, we summarize that we are now dealing with mainly three of the polymerases: alpha, epsilon, and delta. Alpha, which forms a complex to act as a primase enzyme to synthesize primer, and then elongates that primer with the DNA nucleotides. Now, this polymerase alpha, as I said earlier on, it works like a primase you have studied in prokaryotes, which actually needs to synthesize a short RNA primer. And then on this RNA primer, we can add on further DNA nucleotides as per the sequence on the template strand. Now, important point is, after around 20 nucleotides elongation, the polymerase alpha is taken over by polymerase epsilon. So that means the processivity of this polymerase alpha is limited up to 20 nucleotides. Once there are 20 nucleotides, it's no more being able to catalyze the elongation reaction, and then the activity is taken over by another polymerase which we studied. This is polymerase epsilon, and I'm talking about mainly the leading strand. And That activity will be taken up by polymerase delta when it comes to the lagging strand. So we can say that polymerase alpha is working like a primase for both leading and lagging strand. And it has the capacity to incorporate 20 nucleotides, after which, on the leading strand, it's taken up by polymerase epsilon, and on the lagging strand, it is being taken up by polymerase delta. Now, there are other enzymes which are responsible for primer remover. If you remember last time, I told you that primer is not the part of the genetic material. Primer is just being synthesized because of the fact that the DNA polymerase cannot synthesize the chain elongation right from the beginning. So once the replication is being done, we need to remove the primer. And for this, there are a couple of other enzymes present in the eukaryotes where their specific function is to remove the primer. And they are also polymerases, and they have 5' to 3' exonuclease activity. So they cut down the specific primer out of the DNA molecule. Now, here is the picture. I would say that picture is not different if you compare that with the prokaryotes. As I said, the basic process would remain the same, but that's a little bit of more complicated. More enzymes are involved, and more proteins are involved. So if I have to summarize this picture, let's start with the first thing. The first thing was identification of the origin. Once the origin is identified, we know that there are specific enzymes which actually bind to those regions. These enzymes are called helicases. As a result of the binding of the helicase, you can see the hydrogen bonds are being broken down. And as a result of the hydrogen bonds are being broken down, the unwinding takes place. This is the first step. The second step is, as soon as the two strands start to separate, they may have the tendency to rejoin. So in order to prevent that rejoining, we have specific proteins being shown here with the purple balls. These are called single strand binding proteins. Once they attach with the both leading and lagging strand, they prevent the reunion of the two chains. So that means the unwinding is going to process. And as you can see, the DLA helicase goes in this direction. As it moves up, the two chains start to separate, and SSBs, single strand binding proteins, they bind with the leading and lagging strand, thus preventing them to reunite. The third step, which we studied in the last lecture as well, when we do the unwinding of the two strands, there is a topological stress which will prevent further unwinding. And in order to remove that topological stress, we have the enzyme topoisomerase, which actually cuts the DNA and then join again to remove that topological stress, and thereby it provides the opportunity to unwind the rest of the DNA molecule. So wherever there is a topological stress in the DNA molecule, the topoisomerases are going to perform their function in order to remove the topological stress. So this is I would say the initiation of the replication process. Now, once the two strands are separated, there comes some other enzymes like DNA primase, which is alpha. What is it's going to do? It's going to synthesize the primer, and it's going to synthesize the primer on the leading strand as well as on the lagging strand. Then we have the other two DNA polymerases, which are responsible for specifically for the leading and for the lagging strand, and they start synthesizing the new chain. We already have studied that on the leading strand, we have a continuous synthesis going on, and on the lagging strand, we have fragments synthesized called Okazaki fragments, and thereby the process would go on and go on. Ultimately, once the process of replication is complete, there are certain enzymes which are going to cut the primer from the original DNA molecule, and after that, specifically after cutting the primer, on the lagging strand, we have, specific Okazaki fragments which needs to rejoin. And here comes the enzyme, which is DNA ligase. That particular enzyme has the function to join the broken segments and make it one long chain. So now you have two DNA molecules from one DNA molecule, and the parental DNA molecule has been utilized as a template to synthesize two new chains on this. So this is the overall summary of the DNA replication in eukaryotes. Now, as I was saying that during the process of this replication and even during the process of transcription, there are possibilities of DNA damage. And we need to address this issue. And I told you the reason for that. Why? Because if there is any damage being caused to the parental DNA, that means it might be either removal of a nucleotide or addition of a nucleotide or any change in the sequence of the nucleotide. And all these things are being referred to as mutation. If there is a mutation, then this mutation will be preserved in the parental DNA molecule. And then every time, whenever it will undergo the process of replication, it's going to replicate a wrong DNA molecule. And the replication of the wrong DNA molecule would result in the formation of a wrong protein. And so on and so forth, it results in different genetic diseases like sickle cell anemia. So the point is that if there is a possibility of a DNA damage, then there must be a way to bypass the damage or repair the damage. And we have different enzymes and different proteins within the eukaryotic machinery having this specific function. So I'm not going in much of the detail, but I want to just give you an idea about how this DNA damage is bypassed. Now, all organisms need to deal with the problem that will arise when a moving replication fork will encounter a damage, because the replication forks are moving in both directions. And if they reach a point on the template DNA where there is a damage, that means the replication fork will stop, because the damage would result in the replication fork that it cannot run further. Now, if this damage that resulted in the halt of the replication fork, or the replication fork is stopped, that might be very fatal for the cell, and even the cell can die. So in this case, the best way to deal with this situation is to do the repair. If we do the repair of the damage, that means the replication fork will continue. And that repair is being done by the excision mechanism, which I just mentioned to you a couple of slides before that. In some cases, however, the damage may not be repairable. This has to be kept in mind. So in this case, advancing the replication fork may already have unwound the parental strand, and thus preventing excision mechanism from using the complementary strand as a template for repair. So in this case, the excision repair may not yet have had an opportunity to repair the damage. So remember that this damage can be overcome by excision mechanism. But in some cases, the damage may not be repairable because of the advancing replication fork has already resulted in the unwinding of the DNA molecule. And as a result of that, the excision cannot be done. And as a result of that, the damage cannot be resolved. This is a possibility. So it is important for the cell to be able to move the replication fork, as I said earlier, uh, uh, past the unrepaired damage, because of the fact that if the replication fork stops, that results in the long-term blockade of the replication, and the long-term blockage of the replication may lead to cell death. Replication of the damaged DNA provides a sister chromatid as well, like an extra chromatid is being formed. And that can be used as a template for subsequent repair by homologous recombination. So in this case, if excision cannot be done, then there is a sister chromatid produced which will do the same function that it can be used as a template and it will do the subsequent repair of the damage. So replication fork bypass mechanism cannot strictly speaking be considered an example of DNA repair because the damage is left in the DNA at least temporarily. So whatever the damage is, either it can be rectified by excision mechanism or that damage can be rectified by formation of a sister chromatid, but this is a temporarily damage because it has to be fixed, and once it is fixed, the replication fork will carry on and the DNA replication process will carry on. Now, a very interesting comparison when it comes to pro and eukaryotes, I already mentioned that in the start of the lecture that the eukaryotic genetic material is much bigger and much complicated. So the rate of replication in case of the eukaryotes is extremely higher as compared to what you have studied in prokaryotes. Now, in prokaryotes, the replication is normally proceeding at a rate of about 1,000 nucleotides per second. And, last time we studied that on average, the E. coli genetic material is being, roughly duplicated in 20 minutes. So it's say between 20 to 40 minutes. In eukaryotes, the replication take proceed at 50 nucleotides per second. Now, look at the difference. In prokaryotes, it's 1,000 nucleotides per second. In eukaryotes, it's 50 nucleotides per second. That's why it takes a longer time to complete it, and it's completed in almost 60 minutes. Now, one very important thing to understand from this is to justify that why we have multiple origins. Because if we have got only one origin in eukaryotes, it will take ages for the DNA to replicate. So in order to overcome this, process rate, in the eukaryotes, we have multiple origin sites, and that's how it facilitates the process to be completed in a reasonable time compared to the prokaryotes. You can see the prokaryotes is much faster, but just compare the genetic material of the prokaryotes is much smaller than that of the eukaryotes. Prokaryotes taking 40 minutes and eukaryotes taking 60 minutes. But when you compare the size of their genome, it's much, much bigger in eukaryotes. So how that processivity is fast enough in eukaryotes because of the multiple origins and the multiple replication forks. Okay, let's move on to the last topic of our presentation, and that is to talk about mutations. I already have given you a brief overview of what mutation is in the presentation of Introduction to Molecular Biology. But today we're going to discuss that mutation in a little bit of detail. I'm pretty sure that you guys all aware of what mutation is. It can be specifically said as changes in the nucleotide sequence of the genome. It's a broad definition. Any change in the original nucleotide sequence of the genetic material. Why I'm saying genetic material? Because in some cases, RNA could be the genetic material for certain organisms as well. But most of the time, it's DNA. Yeah. So changes in the nucleotide sequence is being termed as mutation. Now, organisms, they have special mechanisms or systems or enzymes which are responsible for the repair, or which are responsible for certain kinds of alteration in the DNA, because mutations are not always welcome. But this is a point to be kept in mind, that mutations are not always harmful. This has to be kept in mind. But most of the time, yeah, we think about mutations are bad. We think about because of the mutation, the final product is changed. But you will come to know later on that not all mutations are harmful. There are certain mutations which might have beneficial impact, but let me talk about it later on. So organisms have a special system of enzymes that can repair certain kinds of alterations in the DNA. Once the DNA sequence has been changed, as I said that it's preserved, the DNA replication will copy the altered sequence. Like if there's a change in the parental DNA, then this change will be transmitted to the newly synthesized DNA. And as a result of that, when it comes to the formation of the protein, that change will be, translated into the wrong protein. So it provides the variation necessary for evolution as well. Like in certain cases, these mutations are naturally occurring process of evolution. Like we see that we have the genetic material coming from our parents and then from their parents, and then there are further generations. So during this process of transmittance of the genetic information from one generation to another generation, there are certain changes which are the part of the evolutionary process. And if you can recall the process of meiosis, you all have studied about that. So let's go to the types of the mutation. We define the mutation on the basis of what type of chromosomes or DNA are mutated into somatic and germline mutation. I think you can understand from this, it's the same concept which we have studied, sex chromosome and the normal chromosome, what we call the autosomes. So somatic mutations are the one that occurs in the cell not dedicated to sexual reproduction. So that means the autosomal chromosomes. So any kind of mutation in the somatic cells, the mutant genes disappear when the cell in which it occurred dies and can only be passed on through asexual reproduction. While in case of the germline mutation, because these are the cells, these are the molecules or the cells which are responsible for the sexual reproduction. So if this mutation occurs, it will be found in every cell descendant from one generation to another, from zygote to which that mutation will be carried on. So that's, if an adult is successfully produced, every one of its cell will contain that mutation. And that's how the mutation disorders are being transmitted from one generation to another generation. We have another classification of mutation which is based on its mechanism. And I'll go in the detail of a few of them quickly. The first type of mutation is called single base mutation or point mutation. I think their names are self-explanatory, and the pictures underneath gives you the idea what it is. Single base mutation, which is called point mutation, is exchange of only one base for another. Like for example, if one purine, adenine or guanine, or one pyrimidine, cytosine or thymine is replaced by another one, this is called point mutation. And if a purine is replaced by a pyrimidine or a pyrimidine is replaced by purine, that's called transversion. So point mutation is a change of one nucleotide. And that one nucleotide change can either be called as transition or it can be called as transversion. Now look at the example. The original is the fat cat ate the wee rat. And there's only one change of the cytosine with the H, and that results in the fat cat ate the wee rat. Now this one point of mutation, one nucleotide mutation, is being referred to as point mutation. The point mutations are continued. That means, because we know that a genetic codon is made up of three subsequent nucleotides. If we go back, and I told you in the intro lecture, these three are being read as one codon. These three are being read as one codon. These three are being read as one codon. So if there is a change of one nucleotide, then this codon is being changed. And you know that when the codon is being changed, that will encode a different amino acid. And that different amino acid will be a small change, but it may give rise to a different protein altogether. And the example, the most relevant example of this mutation is the sickle cell anemia. In sickle cell anemia, the hemoglobin shape is changed from the normal disc shape to a sickle shape. And we know that it's a lethal disease. There is only one mutation of A versus T at the 17th gene of the beta chain of the hemoglobin. And as a result of this, the original code was GAG, which code for glutamic acid, and it's converted to GTG, which code for valine. So one point mutation results in the change of one amino acid. And you can see just change of one amino acid give rise to a drastic change of the characteristic of the hemoglobin, and it becomes sickle shape. And the disease is called sickle cell anemia, which I said is a lethal disorder. Another example of the mutation is the missense mutation. Now, what is missense mutation? Look at the original DNA code of an amino acid sequence. If you start from there, it goes like this: cat, 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 cat. So these are the same genetic code. So you can see that they have same amino acids coming: histidine, histidine, histidine. Now, when you have this A replaced by the C, then this genetic code will become CCT, and the CCT code for proline. So again, you have the replacement of a single nucleotide, and that single nucleotide changes the amino acid. And instead of all histine, it has brought in proline. And as I said, that which may produce a malfunctioning protein. The change of only one amino acid could give rise to a totally malfunctioned protein, as we have seen in the sickle cell anemia. This is another example, just to show you the one that we discussed about sickle cell anemia. You can see here, this is the change, and in place of the normal glutamic acid, there is valine, and you can see the hemoglobin shape is being changed from the normal disc shape to a sickle shape. Now, there are examples of diseases which are caused by these point mutations, which I have discussed with you. Some of them are color blindness, cystic fibrosis, hemophilia, phenylketonuria, and Tay Sachs disease. The other types of point mutation may have some other effects. And as I said, that not all mutations are harmful. So in this case, it might be possible that a change of a codon to one that encodes the same amino acid, and that causes no change in the protein. That's called silent mutation. Remember, we have 61 codons for 20 amino acids. And I told you that there are more than one codons for a single amino acid. So the point is that if there is a small point mutation that changes the genetic code, but that doesn't change the amino acid, that means a correct amino acid is being incorporated, although there was a change of a single base pair. This is being called as silent mutation. Similarly, a change of a single base pair can change an amino acid codon, and it can bring up a codon which is a stop codon. Out of 64, 3 are the stop codon and 61 are for the amino acid. So that small change can bring up a change of a codon from an amino acid to a stop codon. And we know that when a stop codon comes in, it will stop the process of replication. And as a result of that, you will get an incomplete protein. This is being called as nonsense mutation. So you got an understanding that how sophisticated and how detailed these mutations can have an impact on the replication process. So it can have serious effects because it's an incomplete protein. And the reason is because it's not going to do that function. For example, if it's an enzyme and that takes place, that there is a single change results in stop codon, and a stop codon results in an incomplete protein, that means that enzyme is not being capable of doing its reaction. So this is example of the nonsense mutation, where you can see that a change of a single nucleotide base pair results in the stop codon, and instead of a normal protein like this, it stops here. Then you have other types of mutation like insertion mutation and deletion mutation. Again, the name itself are very much self-explanatory. Insertion mutations are those where an extra base pair is being added within the new DNA molecule. Now, how it happens, as I said earlier on, during the process of replication, there are DNA damages. And as a result of that DNA damages, there is a possibility of insertion of certain extra base pairs. So if the original is this one, we have this insertion XLW, which might code for, a different amino acid, and that would result in a different protein. Similarly, in the deletion one, a section of the DNA is lost. Or this can also happen with the DNA damage. As a result of the damage, it might miss a small portion, and as a result of this, one of the base pairs is missing, one of the codon is missing, and then one of the amino acid is missing, and so on. It might give a non-functional protein. These are the example of insertion, mutation, and you can see here, this is the original one. And with the original one, the C is being, there is an extra A added. Now it's not removed. CAT is there, you can see. There is an addition of A. It's not point mutation, remember. So it's an addition of A. Now because of the addition of the A, the reading frame is being changed. Previously, the reading frame was cat, 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 cat. But now here, the reading frame becomes cat, 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 ACA, TCA, TCA, TCA. And that changed the entire sequence of amino acids. Insertion of one nucleotide base pair has changed the whole reading frame, and hence it changes all the amino acid sequences. So this is the example of insertion mutation. Then there are many, many more examples. Like an example of a human disorder which is caused by insertion is Huntington disease. This is one example. In this disorder, the repeated trinucleotide is CAG, which adds a string of glutamine to the encoded protein, and that results in the Huntington disorder. Abnormal protein will increase the level of p53 protein in brain cells, and you know that this p53 protein is involved in cell apoptosis. So it starts the death of the cells, and that's the disorder. This is a normal protein, and this is abnormal protein of the Huntington disease. Deletion mutation, as I said, this time the frame was cat, 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 and here with this cat, the A is being deleted. Now, as a result of the deletion, again the whole reading frame is changed. From there to there, it's cat, 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 then becomes CTC, ATC, ATC, and ATC, and then you've got entirely different amino acids coming in. And that again results in a malfunction protein. So examples of the disease caused by this type of mutation are Cry-do-chat, De-Grouchy syndrome, um, uh, Printzen syndrome, Wolff-Hirschhorn syndrome, and muscular dystrophy. The insertion and deletion mutations involving one or two base pairs or multiple as well. They can have devastating consequences to the gene because, as I said, the whole frame is changed. And as a result of that, the entire sequence is affected. The DNA is read in sequence of three bases, as I was telling you, and if insertion or deletion takes place, the whole frame shift changes. And this frame shift changes not only bring about wrong amino acid, but it might often create a stop codon. And that's how halt the protein and incomplete protein is synthesized. Then there is another mutation called frame shift mutation. Again, the name is explanatory. It's just changing now because of the insertion and deletion. There was a change in the frame, but this is a different type. In this, there's no insertion deletion, rather the entire reading frame is mutated because of the certain factors. So if you look at this original DNA, where it's being read as CAT, TCE, CAC, GTA, and we have these amino acids coming in. This is the normal sequence. Now, once the process of replication is started, there is a frame shift of one DNA base, and that results in abnormal amino acid sequence. So, when the binding takes place for the reading, the binding is wrong, and instead of starting from CAT, it starts from ATT. And then again, whole reading frame is changed, and that is called frame shift mutation. Now you can see that there is no insertion or deletion. The base pair is there, but the reading frame has been disturbed, And as a result of this, we have entirely different set of amino acids coming in and resulting in uh, malified or malfunction protein. Then the other types of mutation include duplications. Duplications are sort of doubling of a genome. During meiosis, we know that crossing over takes place between sister chromatids that are out of alignment, and as a result of that, it can produce one chromatid with a duplicated gene, and other having two genes with a deletion. So in this case, there are certain genetic disorders related with this type of mutation. An example of that disease is DMI, which is called myotonic dystrophy. This is the example of duplication mutation. When the crossover takes place, one part of that chromosome was actually being added in this chromosome, giving an extra length to this and a shorter length to this chromosome, and that is being called as duplication mutation. Then you have translocation mutation as well. These translocation mutations are actually the transfer of a piece of a one chromosome to a non-homologous chromosome. In this case, the transfer was to the homologous chromosome. But in this case, one part of the chromosome is being transferred to a non-homologous chromosome. This is being called as translocation, and translocations are often reciprocal. That means the two non-homologous will swap segments. This is another example of this. You can see that this is chromosome 4, this is chromosome 20. These are non-homologous. But there is a part of that chromosome 4 which is being added in chromosome 20, and that results in translocation. Right? Translocations can also alter the phenotypic behavior of the organism in different ways. For example, the break may occur within a gene, results in the destruction of the function or creating a hybrid gene, a duplicate hybrid gene. The translocated genes may also come under the influence of different promoters and enhancers which are required for DNA replication, so that their expression is altered. Then we have inversion. In the inversion mutation, an entire section of DNA is reversed, inverted. A small inversion may involve maybe only few base pairs within a gene, while in case of the longer inversions, it may have larger regions of the chromosome involving multiple genes in this. So, the inversions can be at the smaller scale, or it can be at the larger scale. As you can see, the original and insertion being shown here. This is the classical example of inversion. This is ABEDCFG, so become ABCDE. This particular portion is being inverted, you can see. And as a result of that, the mutation takes place. Then there are mutations called suppressor mutation. The name itself indicates that these mutations suppress. What actually happens in this? It results because of partially or completely masking the phenotypic expression of a mutation, but occurs at a different site from where it causes the suppression. So this sort of suppression mutation will mask, it will not be shown in phenotypic expression. Maybe intragenic or maybe intergenic, like in the genes or between the genes. It is used particularly to describe a secondary mutation that suppresses a nonsense codon created by a primary mutation. So this type of mutation, suppressor mutation, primarily affects the nonsense codons, which are being also called as the stop codons. So these are the types of the mutation in short: deletion, duplication, inversion, insertion, translocation. All these types are being summarized here. Finally, we end up the lecture. This is naming genes. I'm not very much interested in this, but just to let you know that all the genes of the human have been characterized, and that project was called the Human Genome Project. And all the genes of the human, they have been listed, they have been identified, they have been given their names, their sequences have been identified, and that's a major milestone towards the understanding of the genetic material of the human body, and that's gonna be very, very effective for any sort of treatment for all the genetic disorders and genetic mutations. How the genetic conditions are named, I'm not very interested in this. We'll just go through it because these are generally being named on the basis of the scientist, or maybe on the basis of the shape or symptoms or geographic area and all that kind of stuff. So that's not pretty much interested. So we finally ended up the lecture where here we have the references if you want to go through that, or you can refer to the books that I recommended you earlier on for the understanding of the topic. So I'm going to stop this presentation here, and I am going to ask for you guys if you have got any question, you can just raise your hand and ask the question. Now over to you guys. Yes, Mahnur.
अस्सलाम वालेकुम सर।
जी वालेकुम अस्सलाम.
सर ये आपने क्वेश्चन पूछा था, बताया था जस्टिफाई व्हाई वी हैव मल्टीपल ओरिजिन इन यूकेरियोट्स। सर कैन यू प्लीज एक्सप्लेन दिस अगेन?
Yes. What I was saying is, like, when you compare the size of the genetic material in a prokaryote and a eukaryote, there's a huge difference. Prokaryotic genetic material is much simpler. It may have few hundreds to few thousand genes. While in case of the eukaryotes, the size of the DNA, the size of the genetic material is much, much bigger, and it has got thousands and thousands of the genes. So we have seen that the speed with which the nucleotides are, incorporated, and the speed with which the replication is being done in prokaryotes, and if we compare that with the eukaryotes, if we go with the same speed, it will take much longer time for the eukaryotic DNA to replicate. Now, in order to overcome that, there are multiple origins. That means there are multiple replication forks. So that means the replication of the DNA on the eukaryotic DNA starts at multiple sites. Thereby, it completes the entire process of replication within the time of 60 minutes, which I mentioned on the slide. I think it's clear to you now.
यस सर, थैंक यू सो मच।
Welcome. Nimra Mahmood. Yes.
Assalamu alaikum, sir.
Walaikum assalam.
Can you please explain suppressor mutation?
Yeah, just hold on. I'll share the screen with you guys again. Yes, I'm going to share the screen, and then you can ask about the specific question that you have. Yeah. What is actually your question about the suppressor mutation? If you can elaborate to me.
सर जैसे कि आपने कहा कि फिनोटिपिकली एक्सप्रेशन होता है इसका, कि ये या तो इंट्रोजेनिक होता है या फिर इन द सेल होता है, लेकिन मुझे समझ नहीं आ रहा कि किस तरह से सप्रेस होता है, या तो इसमें जो सीक्वेंस है वो सप्रेस हो जाता है। आप थोड़ा सा ये मुझे
Okay.
डिस्क्राइब भी थोड़ा गाइड कर दें।
Okay. Okay. First of all, let me clarify to you that suppressor mutation is not something which is very significant when it comes to mutation, or when it comes to genetic disorders. Like, what is actually happening is that a particular part of the gene is suppressed. Suppressed means it's not being replicated, and it's not being translated into a protein. Now, what I actually was saying, that it masks phenotypic expression. When there is a gene expression, there are two phenomenon. Either it is genotypically expressed, and sometimes it's phenotypically expressed as well, like in certain genetic disorders. Most of the suppressor mutations they mask phenotypic expression. That means you won't see any phenotypic expression, you won't see any change visible, which actually is resulted because of the suppressor mutation. And the reason for that is that sometimes this mutation takes place in the intergenic region. If you remember, there was the region between the two genes and the genes itself. So intergenic region was the region between the two genes. So if it's between the intergenic region, it's not going to phenotypically express and it's not going to affect anymore. While in case of the intragenic, this is particularly the gene region. But this might be the case that there is any mutation which is not phenotypically expressed. So that's why they are not pretty much related to any clinical condition, and that's why we haven't mentioned any clinical condition with this kind of mutation. Is that clear?
Yes, sir. Thank you.
Yes, any more question, guys? Okay, so we are going to stop here, and inshallah, I'll share the recording with you guys, and the presentation has already been shared with you. Go through it, and if you have got any question, you can come back to me at any time. Thank you very much. Khuda Hafiz.